John is affiliated with Virginia Bioinformatics Institute. And so today he's going to be speaking to us about the eukaryotic cell cycle, molecules, mechanisms, and mathematical models.
Thank you very much. It's a great honor and privilege for me to give this keynote address, and I hope I can keep you interested for the next 55 minutes or so. And we'll have plenty of time for questions at the end. So I want to talk about the molecular mechanisms that underlie the control of progression through the eukaryotic cell cycle. Before I get started, let me acknowledge that most of the work I'm going to talk about is funded by the Institute of General Medical Sciences at NIH. So the cell cycle is the sequence of events by which a growing cell replicates all of its components and divides them more or less evenly between two daughter cells, so that the daughter cells inherit the information and the machinery necessary to repeat the process. And in all cells, the most important components that have to be replicated and then partitioned to the daughter cells, the most important molecules are, of course, the DNA molecules, the genome. And in eukaryotic cells, the process of replication of the genome and then partitioning of the replication products to the two daughter cells, they occur in two temporally distinct phases of the cell cycle. And these are usually called S phase and M phase. So S phase is the phase of DNA synthesis, when the single-stranded DNA molecules are replicated to form two double-stranded DNA molecules. And so at the end of S phase, each chromosome is replicated, and the two sister chromatids are held together by proteins called cohesins. They're glue that hold the two sister chromatids together. Then when replication is complete, eventually the cell enters mitosis, and in the process of mitosis, these sister chromatids have to be separated. So first the cell builds a mitotic spindle. The spindle captures the replicated chromosomes and aligns them on the metaphase plate. And when all the chromosomes are properly aligned, then the glue is dissolved and the two sister chromatids are pulled to opposite poles of the mitotic spindle, and the cell divides, and you have two new cells back at the beginning of the cycle. Now, why do we want to study the cell cycle? Well, that's an easy question to answer. As we know, all living organisms are made of cells, and all cells come from previously existing cells by the process of cell growth and division. So the cell division cycle is, in my opinion, the most fundamental process that occurs in living organisms. So of course we want to study it, we want to understand the molecular mechanisms that control progression through the cell cycle, make sure that everything happens decently and in order. We should realize that there are many human diseases that are disruptions of the proper controls of the cell cycle, especially cancer. Cancer is a family of diseases that are all concerned with misregulation or dysregulation of cell growth and division. I'm going to talk about the control of cell growth in budding yeast cells on the left, and you might ask, if you're interested in cancer, why don't you study the control of cell division in a cancer cell? And that's a little harder question to answer, but I would just use an analogy. If you want to become a cell mechanic, which car would you start with? I think it's reasonable, if you're a beginning mechanic, to buy an old Beetle and fuss around with that, okay? It's a relatively simple machine, and you can fix it, you can damage it, you can repair it, you can learn a lot of car mechanics, and you don't want to be messing with this, I think that's an Audi R8 or something like that. Maybe that's an $80,000 machine, I don't know. You don't want to tinker with that baby. But you know, these are basically the same machines. They work on the same principles, and you can learn most everything you need to know about auto mechanics by studying the Beetle. Okay, then you have to go and become a specialist on race cars. So we're going to learn everything we can about the Volkswagen Beetle of the eukaryotic kingdom, which is the budding yeast. Let me step back, talk about what I consider to be the four most fundamental properties of the cell cycle. These are the things that almost all cells share, and that certainly we have to understand. If we think we understand the molecular regulation of the cell cycle, it should explain these basic properties. And I should also say, this is biology, okay? So almost any rules that you lay down have lots of exceptions, okay? And most of you here will be able to think of all kinds of exceptions to the rules I'm laying down. Don't let that, sink the boat. These are pretty general rules, I think. First of all, an alternation of DNA synthesis and mitosis. The job is to replicate the DNA and then separate the two sister chromatids. And then you go through another round of DNA synthesis and then through another mitosis. So it's important for the cells to alternate DNA synthesis and mitosis. And if they don't do the alternation, if they skip a phase and repeat a phase, they run into trouble. This alternation of DNA synthesis and mitosis is controlled by checkpoints. And by checkpoints, the cell physiologist means that at certain phases in the cell cycle, the cells have to make a decision. Okay, so suppose a newborn cell is exposed to ultraviolet radiation, which damages the DNA. Then the last thing that cell wants to do is go into S phase and replicate the damaged DNA. What the cell has to do is repair the damage first and then go into DNA synthesis. So it needs a checkpoint that checks for DNA damage, and if the DNA is damaged, that checkpoint prevents progression to the next phase of the cell cycle, S phase, until the damage is repaired. Then the checkpoint is lifted and the cell can go on. Okay, that's a very crucial checkpoint in the cell cycle. Another checkpoint is at the metaphase to anaphase transition, because the whole purpose of metaphase is to align the replicated chromosomes on the metaphase plate, and that takes some time. We have 46 chromosomes. It takes a while for all 46 to get properly aligned on the metaphase plate. And if even one of the 46 chromosomes is not yet aligned on the metaphase plate, there's a checkpoint that sends a signal that prevents progression from metaphase to anaphase and the separation of the chromosome. So these checkpoints work to maintain the integrity of the genome. And we have to understand how what's the molecular mechanism of these checkpoints. Thirdly, I'm emphasizing the cycle of DNA synthesis and mitosis and division, but there's also a cycle of growth that the cells are undergoing all the time. As the DNA is being replicated and divided, the cell is growing, just in terms of total cell mass, proteins, organelles, membranes, everything's increasing. And on average, the cell has to go through one round of DNA replication and division every time the cell mass doubles. But those are two entirely different processes, but they have to be coordinated. In other words, on average, the mass doubling time of a culture has to be the same as the cell cycle time, the time it takes to replicate the DNA and divide. You have to balance growth and division, or else cells will get too large or too small. And finally, the fourth characteristic is that this progression around the cell cycle has to be very robust. The cell can't afford to make mistakes, okay? It has to be done properly. It has to be robust. And yet, yeast cells are very, very small, and they just don't have very many molecules, which means it's quite a noisy environment from a molecular point of view. And yet, in the face of this unavoidable molecular noise, this control system has to work with complete fidelity. These are the four things that we'd like to understand. Okay, next we have to know a little bit about the underlying molecular biology. The events of the cell cycle are controlled by a family of protein kinases called cyclin-dependent kinases, CDKs. The cyclin-dependent kinases have no activity on their own. They must find a partner, a cyclin protein partner, okay? And in budding yeast, there's only one CDK to worry about. It's just one kinase subunit. There's lots of kinase subunits. The budding yeast cells are full of the kinase subunits. They can't do anything until they find a partner. Early in the cell cycle, the kinase subunits partner with a cyclin called Clin2, and that partnership kind of coaxes cells to leave G1 and kind of move towards DNA synthesis. But in order to do DNA synthesis, the cyclin-dependent kinase has to partner with a different cyclin called CLIB5. That's the primary partnership that drives DNA synthesis. And then towards the end of the cycle, the kinase subunits partner with a third cyclin called CLIB2, and that drives the cells from G2 into mitosis and up to metaphase. And high activity of the CLIB2 dependent kinase prevents the transition from metaphase to anaphase. So from the end of G1 through S, G2, and M, we have high activity of cyclin-dependent kinase partnered with one or another of these cyclic proteins. In order to exit from mitosis and return to G1, we need a different suite of players. The first one you need to know is a protein called CDC20, which is a regulatory protein that regulates a machine called the APC, the Anaphase promoting complex. And what this machine does, when it's active, this machine causes the dissolution of the glue that's holding the sister chromatids together, so it allows anaphase to occur. That's why it's called the Anaphase promoting complex. It dissolves the glue and the sister chromatids can separate, the cell goes into anaphase, but the CDC20 APC also destroys all these cyclins, the CLIB2 and CLIB5 and all the other B-type cyclins. They're degraded by the APC. And that releases this inhibition that allows the cells to exit mitosis, go back into G1. They're held in G1. Cells are held in G1 by two other proteins. A protein called CDH1, which does basically the same thing that CDC20 does. They're kind of brother and sister. And another protein called SYK1, which is a stoichiometric inhibitor of cyclin-dependent kinase complexes. These are just important proteins in G1 of the cell cycle that keep the activity of cyclin-dependent kinases alive. So that's the organism, budding yeast, the physiology that we're interested in, the basic molecular components. Now, how do we put these components together to try to understand the general principles of cell cycle regulation in budding yeast? Now, here's a proposal. This is a hypothetical or a proposed wiring diagram for how these components interact with each other, and it's proposed that in some sense, in some way, this wiring diagram explains everything we need to know about the budding yeast cell cycle. More or less in the same way that if you could get hold of the wiring diagram for your cell phone, you could figure out how your cell phone works. Well, what does a wiring diagram look like? The icons are molecular species. So this is the dimer between cyclin-dependent kinase and CLIB2. This dimer is, remember, the cyclin-dependent kinase is always present in excess. So what controls this dimer is the supply of CLIB2. So here's the synthesis of CLIB2 driven by some transcription factor, and here's the degradation of CLIB2 driven by CDC20 and CDH1. So all the solid arrows are chemical transformations: synthesis and degradation, phosphorylation and dephosphorylation, association and dissociation. All of the dashed arrows are catalytic activities. So one component of the system is a catalyst for a reaction someplace else in the wiring diagram. That's indicated by the dashed arrows. Okay? So this hypothetical wiring diagram is based on years and years of molecular biology from the top labs around the world, trying to sort out what these components are doing inside a yeast cell. And our job as systems biologists is to try to figure out from the wiring diagram how the control system works. So first I want to talk about deterministic modeling of this wiring diagram, and then I'll talk about stochastic modeling and noise in yeast cells. The deterministic modeling has been done for a number of years now with my primary collaborators, Bela Novak and Kathy Chen, and two former students, Attila Cekashnaj and Andrea Ciliberto, who did a lot of this work that I'm going to describe. So it's a complicated wiring diagram, and I want to try to simplify it. I want to try to focus in on what we consider to be the really crucial molecular interactions that govern this system. And first of all, I want to draw your attention to the relationship between CLIB2-dependent protein kinase. Remember, that's the one that drives mitosis. But we could also put CLIB5 here as well, the one that drives DNA synthesis, because it's controlled in a very similar way. But let's just talk about CLIB2. So CLIB2 is being synthesized and being degraded. I'm going to assume, for simplicity, that CLIB2 is synthesized at a constant rate. That's not true, but we'll just assume it for the time being, for simplicity. There's a constant rate of synthesis of CLIB2, but a variable rate of degradation. The rate of degradation is controlled by the activity of CDH1. But the activity of CDH1 is controlled by its phosphorylation state. CLIB2 is a protein kinase, and it can phosphorylate CDH1 and inhibit the activity of CDH1. So that's a kind of curious interaction between these two components, because CDH1 is trying to destroy CLIB2 and destroy the activity of the kinase, but the kinase is trying to phosphorylate CDH1 and destroy the activity of CDH1. So these two molecular components are antagonists, they're enemies. Okay? That's their relationship with each other. Now, what does this antagonism imply from a dynamical point of view? Well, let me abstract this part of the complicated mechanism as a simple little motif: synthesis and degradation of a protein X and phosphorylation and dephosphorylation of a protein Y. And write a pair of differential equations that describe the rates of these reactions just by standard biochemistry. So here's the synthesis of CLIB2 driven by some transcription factor S. Here's the degradation of CLIB2 catalyzed by CDH1. And here's the phosphorylation of CDH1 by the cyclin-dependent kinase. And here's the dephosphorylation of CDH1. Now, how do we solve these differential equations? Well, I want to just draw pictures to solve them. The picture I want to draw is a picture that is a graph that plots the steady state activity of the cyclin-dependent kinase, that's species X in this set of differential equations, as a function of a parameter, and the parameter is the ratio of the activity of the transcription factor to the activity of the phosphatase that appears. What's curious about this diagram, because of the antagonism between the cyclin-dependent kinase and CDH1, the system has two different stable steady states. It has a stable steady state with very low activity of the cyclin-dependent kinase. It's low because CDH1 is very active, and CDH1 keeps destroying the CLIBs. But it has another steady state with high activity of the cyclin-dependent kinase. It's high because now CDH1 is kept phosphorylated and inactive, and CLIB2 can accumulate. So there are these two different steady states, but there's a region of coexistence of these two steady states. That's called the bistable region. And this works like a little toggle switch. If we start in the off state and increase the activity of the transcription factor S, then we can drive the cell to switch from the off state to the on state, to make an irreversible transition and to turn on. Now the cyclin B-dependent kinase activity is high, the cell can do DNA synthesis and go into mitosis. But then it'll block at mitosis because it's at the upper steady state. In order to exit from mitosis, we have to reduce this parameter. We have to go around the bend on the other side, so maybe we have to activate the phosphatase A and drive the parameter down, and we can flip the switch back. Now that's the basic idea. The molecular antagonism creates a bistable switch. The two states of the switch correspond to the two different fundamental phases of the cell cycle. G1, which is uncommitted to DNA synthesis and mitosis, and SG2M, where the cell is in the midst of doing DNA synthesis and mitosis. And the cell has to flip back and forth between these two states and has to go around and around this cycle. Each turn around this cycle is one cell cycle. The question is, what are the mechanisms that flip the switch on and off? So let's go back to the wiring diagram and see what we can see. Here's the bistable switch, and there's another component, CLIN2, in this diagram. And CLIN2, dependent kinase, has the property that it can also phosphorylate CDH1 and inactivate CDH1. So that's why I put this little negative sign here, because CLIN2 is an inhibitor of CDH1. Since CLIN2 is an enemy of CDH1, it's a friend of CLIB2. Got it, right? CLIB2 and CDH1 are enemies. But, unfortunately, right, CLIN2 helps out CLIB2, but CLIB2 does not return the favor, because CLIB2 comes down here and phosphorylates the transcription factor that's driving the synthesis of CLIN2 and inactivates the transcription factor. So when CLIB2 comes up, it turns off the production of CLIN2, and CLIN2 is a very unstable protein. Rapidly it goes away when you shut off its synthesis. So we have this curious kind of a motif here, where we have the molecular antagonism that creates a bistable switch, but the bistable switch is embedded in a minus, minus, minus negative feedback. Well, what's that doing? I want you to remember this. I want you to remember that CLIB2 inhibits CLIN2. CLIB2 knocks down CLIN2. So when CLIB2 is high, CLIN2 has to be low. Okay? We need that. Here's the bifurcation diagram for the CLIB2 activity as a function of the CLIN2 level. Now, remember that this is like the bifurcation diagram that I showed you earlier, except it's only half of a toggle switch instead of a whole toggle switch. That's important. We just have half of the toggle switch. And CLIN2 is going to be the parameter that I want to use to flip the switch. Now, how am I going to do that? If there's no CLIN2 in the system is bistable. It can be up here or it can be down here, but at the beginning of the cell cycle, it's sitting down here. That's the G1 state of the cell cycle. Remember, CLIN2 is an enemy of CDH1, and CLIB2 is an enemy of CLIN2. So I should really draw the diagram something like this. When CLIB2 level is low, CLIN2 level can be high. But as CLIB2 increases, CLIN2 level has to decrease. That's what that blue line is telling me. But now if the cell starts here, it was in G1, and it starts making CLIN2, then CLIN2 drives the cell past the switching point. The switch flips to the upper steady state. Now CLIB2 level is high, and CLIN2 goes away, and we end up on the upper steady state. So we embed a little toggle switch in a negative feedback loop, and the negative feedback loop can cause the switch to flip, but then the trigger goes away, and we end up on the upper steady state. And that's just the transition from G1 through this up jump here triggers DNA synthesis, and then the cell goes into mitosis. Okay? So now we're up at the other stage of the cell cycle. And we have to figure out how this control system flips back. So we go back and look at the diagram. Here's our toggle switch. And what's all this stuff doing up here? This protein, CDC14, is a phosphatase, and if you follow the dashed arrow, it's a phosphatase that takes the phosphate group off of CDH1 and activates CDH1. So CDC14 is a friend of CDH1. And all of this business up here tells you that high activity of CLIB2-dependent kinase eventually causes the activation of CDC14. But CDC14 then activates CDH1, and CDH1 kills CLIB2. So we have this kind of a motif. Here's the bistable switch. It's embedded in a loop, plus, plus, minus. So that's also a negative feedback loop. A toggle switch embedded in a negative feedback loop. And what does that look like? If CDC14 level is low, here's our system with two stable steady states, but now we're up here. We're in the on state. We're stuck at mitosis, and we want to exit from mitosis. How are we going to do that? Well, remember, CLIB2 activates CDC14. So when CLIB2 level is high, CDC14 level wants to be high. When CLIB2 level is low, CDC14 level wants to be low. So if we start up here and activate CDC14, then the switch will flip off. This drop in kinase activity drives cell division, and then CDC14 goes away, and we return to that steady state. So now we flip the other direction. Okay? So what I'm trying to say is that big complicated diagram is to create essentially this picture. A little toggle switch embedded in two negative feedback loops. One negative feedback loop flips the switch on, and the other negative feedback loop flips the switch off. And when I put the two bifurcation diagrams back to back, we can now see the cyclic nature of the cell cycle. From G1 to S phase, go into mitosis, and then exit from mitosis, anaphase, telophase, cell division, and back to G1. Okay, so that's the cell cycle on the bifurcation diagrams. Now, is any of this true? Or is this just a story that I like to tell? Well, we need some experimental evidence. Let's suppose we knock out the negative feedback loops and just concentrate on the bistable switch. If we could do that, then in experiments we ought to be able to arrange a cell which is either in the low steady state and it looks like a G1 phase cell, or it's stuck in the upper steady state and it looks like a metaphase cell. So can we do that experimentally? A very elegant trick was devised by Fred Cross, which I'm going to explain in the next slide, but I've got to prep you for it so you can understand, because it's a pretty tough experiment. What Cross did, he knocked out Clin2 and those kinds of cyclins, and he expressed a cyclin called Clin3 with some special properties, which I will explain. And he was able to control the activity of Clin3. And what I'll show you in this experiment, if he just produces a little bit of Clin3 and then turns Clin3 off, the cell tries to move out of G1, but then it changes its mind and goes back to G1. If he just produces a little bit of Clin3. But if he produces more Clin3, then he can get the switch to flip and the cells will go into mitosis. Okay, so that's the test of whether the system is bistable or not. You just do a transitory perturbation. If you do a little perturbation, the cells come back to where they were. If you do a big perturbation, they go to an entirely different state. At the end of the experiment, the cells are under identical conditions, but some cells are down here and some cells are up there. So let me show you how Fred did this experiment. Okay, this is a bit of yeast genetics. It's the marvels of what you can do with yeast. First of all, there are 3 Clin-type cyclins, and you knock them all out. You knock out Clin1, Clin2, and Clin3. There's no Clins. It can't start the cell cycle. That would be an inviable mutation. Then he puts the Clin3 gene under control of a galactose-inducible repressor, so he can make Clin3 by adding galactose to the growth medium. And then he can stop making Clin3 by transferring the cells from galactose to glucose. They'll immediately shut off the production of Clin3, and Clin3 is a very unstable protein, so as soon as you shut off its production, it goes away. Finally, we got to get rid of CDC14. Remember, CDC14 was the guy on the other side of the picture. We have to get rid of CDC14. He does that by using a temperature-sensitive allele of CDC14. So at 37 degrees centigrade, CDC14 is inactive and the cells can't exit from mitosis. That way you can trap them in the upper state. This is a complicated genetic experiment, but the important thing to realize is when the cells are growing in glucose at 37 degrees centigrade, they don't have any Clins and they don't have any CDC14. So that's the neutral condition that I was telling you about. Now, here's the experiment. First of all, he grows cells in galactose at 23 degrees. In galactose at 23 degrees, they have Clin and they have CDC14, so they're perfectly happy. They live in galactose at 23 degrees. So this is an asynchronous population of yeast cells. 45% of the cells are unbudded in G1 phase of the cell cycle. So 55% of the cells are budded. They're in S, G2, or M. They're going through the cycle. It's an asynchronous population. And if you do a gel, you find that there's plenty of CLIB2 cyclins because there's plenty of cells doing mitosis. That's perfectly fine. These are viable cells. Then he shifts to raffinose at 23 degrees, and now there are no Clins at all. So cells finish the cell cycle, they come into G1, because they have no Clin cyclins, they can't leave G1. So they pile up in G1. Now 88% of the cells are unbudded, and you can't see any CLIB2 because all the cells have cleared out and none of the cells have gone back into the cycle. So there's no CLIB2 around. Those cells are going to be dead if you hold them like that for long. After he catches them all in G1, then he switches them to raffinose plus galactose for no time, or a half an hour, or 1 hour. When they're growing for this short period of time in galactose, what do? Team? they synthesize Clin-3. Okay? If it's a half an hour, they synthesize a little bit of Clin-3. If it's an hour, they synthesize more Clin-3. And then, at the end of that time, okay, so you make some Clin-3, more and more Clin-3, and then he shifts the cells to glucose at 37 degrees. That's neutral. Shifts them into neutral, holds them there for 2.5 hours, and he asks, where are you? Are you in G1? Are you in SG2N? The cells that didn't get any Clin-3 are, of course, they're still in G1. 85% unbudded, no Clib2. They're still in G1, of course. They didn't get perturbed. The cells that saw Clin-3 for a half an hour are still in G1. 86% unbudded, just a little smudge of Clib2. Maybe a few of them were able to get into the other phase of the cell cycle. But if you made an hour's worth of Clin-3 and then you shift into neutral, now 75% of the cells are budded and there's lots of Clib2. That means a lot of the cells have made the transition and they're now in the upper states. But all the cells, at the end, all the cells are under exactly the same conditions. They're all in neutral. They have no Clins. They have no CDC14. And yet you've divided the cell population into two populations. Some of the cells are in a stable G1 state, and some of the cells are in a stable SG2M state. Okay? So the system really is a bistable system. And you can flip them by perturbation with Clin-type cycle. So that's the experimental proof of bistability on this side of the diagram. What about the other side of the diagram? Are you having fun with this yeast genetics? It's kind of weird. You have to get used to it, but it is so powerful, extremely powerful. Now, let's see what we can do on the other side. Now, we've knocked out CDC14, so we can't get out here. We can't go around this loop because we don't have any CDC14. But there's another way we could possibly induce the transition, and that is we're going to give the cells a little burst of CDH1. But it's a special kind of CDH1. It's a CDH1 that cannot be phosphorylated by Clib2. So it's said to be constitutively active. CDH1CA, constitutively active. That means this CDH1 can't be killed by Clib2. Good. So if we put this CDH1 into the system, then it'll drive Clib2 down a ways. But if we can manage to just give a burst of CDH1, if we give a small burst of CDH1 and drive it down a ways, it will turn around and come back to the end state. If there's two stable steady states, if you give a little bit, you go a ways and then you go back to where you were before. And if you give a little bit more, then you continue on and you flip to the other state. It's the same experiment, just done on the other side of the diagram with a different set of genes. Is the system bistable or not? At the end of the experiment, the cells are going to be in exactly the same state, but some of the cells are going to be down here, and some of the cells are going to be up there. Cool experiment. This one was designed by Frank Ullman at Cancer UK, together with Bela Novak. And here's the genotype. First of all, CDC20 is on a methionine repressible promoter. So by growing the cells in methionine, they have no CDC20, which means they can't exit from mitosis. They get blocked in metaphase. So we want to do that. We want to block them in metaphase first. Then he puts the constitutively active CDH1 gene under the control of the galactose promoter so he can turn it on and off. So you can turn it on with galactose. It's a little hard to turn it off because CDH1 is not an unstable protein. So you can make it, but once you've made it, you can't get rid of it, unfortunately. But he's got to kill it. The way he kills it is by having a temperature-sensitive gene called CDC16, which happens to be an essential component of the APC. And remember, CDH1 and the APC work together. You got to have both of them to degrade cyclins, okay? So first you make the constitutively active CDH1 with good APC, and you degrade cyclin, and then you raise the temperature, CDC16 unfolds, the APC no longer works. Cool experiment. So we're going to add galactose at 23 degrees centigrade to turn on CDH1, and then we're going to shift the temperature to 37 degrees centigrade to turn off CDH1 activity. So here's the experiment. First of all, you repress CDC20 so the cells pile up in mitosis. You can tell the cells have piled up in mitosis because they have lots of cyclin D. They don't have any of this SYK1, the kinase inhibitor. And here's what the nuclei look like. Can you see that? Those are metaphase nuclei. Okay, the spindle is lined up, the chromosomes are all lined up on the spindle. At time 0, you add galactose, so you turn on CDH1, constitutively active, and you can see now, over the next 30, 40, 50 minutes, the cells accumulate a lot of constitutively active CDH1, and what happens to cyclin B? Cyclin B gets degraded and goes away. It looks like these cells are exiting mitosis, right? That's what happens when cells exit mitosis. CDH1 degrades cyclin B, and at 50 minutes, this is what the cells look like. They look like interphase cells. They look like they're almost done dividing. But then, at 50 minutes, they raise the temperature to 37 degrees centigrade, and lo and behold, cyclin B comes back again, prematurely, and the cells return to metaphase, just like predicted by the mathematical model. On the other hand, if you wait 10 minutes longer and you raise the temperature to 37 degrees at 60 minutes, then it's too late. This doesn't happen. Cyclin B doesn't return. CKI doesn't go away. The cells remain in interphase. They don't go back to mitosis. So there's this time point again. If you do it before the transition is complete, the cells can go back to where they were. If you wait a little bit longer, the cells go forward to the other steady state. So on both sides, this control system is working like a bistable switch. That for me is the experimental justification or confirmation of the predictions of the model. And these were true confirmation, these were true predictions, right? These experiments were designed and carried out after we published the mathematical model. And these particular experiments I explained were done specifically to test this prediction of the mathematical model. Once you see how this works, immediately you see why cells alternate DNA synthesis and mitosis, because they're forced to go around that loop. And that loop alternates DNA synthesis and mitosis. How do checkpoints work? Well, if the cell has damaged DNA, what does it want to do? If the DNA is damaged, it has to prevent this transition, which is going to drive the cells into DNA synthesis. And how do you prevent that transition? You prevent that transition by somehow preventing the accumulation of CLIN2 activity. And there are many ways to do that, but that's exactly how that DNA damage checkpoint works, by interfering with CLIN2, the starter kinase. On the other hand, the checkpoint that checks for chromosome alignment on the metaphase plate, if the chromosomes aren't aligned yet, if you're having problems aligning your chromosomes, you have to inhibit this transition. And of course, the way to inhibit this transition is to prevent the activation of CDC14. And that's the way that checkpoint works. And you can go back and see in the wiring diagram, that's exactly where the checkpoint operates. And finally, balanced growth and division is controlled by the fact that small cells are not able to make CLIN2. Cells have to grow to a sufficiently large size before they can make CLIN2 and do this transition. So cell growth activates CLIN2 production and coordinates the DNA division cycle with the cell growth cycle. So that's the end of the deterministic modeling. And now the question is, this all seems to work well with ordinary differential equations and bifurcation diagrams, but will it still work in the inevitably noisy molecular environment of a tiny yeast cell? And how tiny are yeast cells? They have a volume of 40 times 10 to the minus 15 liters. That's pretty small. How small is it? How many molecules are in a yeast cell? I pulled these numbers off the web because high throughput studies have counted molecules in yeast cells. And if you look at the counts for proteins, you see the average number of proteins of different cell cycle genes in an asynchronous population of yeast cells. So they're all different phases of the cell cycle. You grind them up, you measure the proteins, you quantify them. And the average number per cell varies from a couple of hundred to a couple of thousand. Well, that's what I would have guessed. The average number of messenger RNAs, this is a surprise. According to the web, there's only 1 or 2 molecules of messenger RNA of any particular gene in an average yeast cell. That seems to be an awfully small number. The half-lives of proteins vary all over the map, from very unstable proteins like Cyclin-2 with a 5-minute half-life to very stable proteins like CDC28. That's the gene that encodes the cyclin-dependent kinase, a very stable protein. According to the web, mRNA molecules are pretty stable. They have a half-life of 10 to 20 minutes, and the cycle time is 90 minutes. So that's a pretty long half-life and low mRNA number. I'm going to show you this causes real troubles. You know not to believe everything that you read on the web, right? And there are very good reasons why we shouldn't believe these mRNA numbers. Let's see what the consequences of these numbers are. If you have a simple birth-death process for a molecule, so this is protein synthesis and protein degradation, then a physicist will tell you that the rule of thumb is the average number of protein molecules is going to be the rate of synthesis divided by the rate of degradation, and the variance of the protein number is equal to the mean. The variance is equal to the mean. And that means that the coefficient of variation of number of protein molecules goes like 1 over the square root of the average number. And if the average number of protein molecules is on the order of 1,000, then the coefficient of variation is on the order of 3%. No problem. Nothing to worry about. We can live with 3% fluctuations up and down in protein numbers if we've got thousands of proteins in the cell. That's not going to cause a problem. However, proteins aren't made that way. Proteins aren't made by a simple birth and death process. Proteins are encoded by mRNAs, and mRNAs are also fluctuating due to synthesis and degradation. And if you take into account the coupling between transcription and translation, it's an entirely different rule of thumb. The rule of thumb is more complicated. It depends on the average number of protein molecules. It also depends on the average number of messenger RNA molecules. And it depends on the half-life of messenger RNA compared to the half-life of protein. We have all those numbers from the table. When we plug them into the rule of thumb, we now find the expected coefficient of variation is 70%. That means if these numbers were true, the number of protein molecules would be fluctuating all over the place. Plus or minus 70%, 100%. You'd have no idea how many proteins you have. There'd be way too much noise in the system for anything to work. So this is what really stimulated us to try to build stochastic versions of our deterministic models to try to resolve some of these issues. Well, how variable is the yeast cell cycle? Here's some simple things we can measure. We can measure the cycle time. Oh, by the way, it's called budding yeast because it makes little buds and it divides asymmetrically. So the daughter cell is smaller than the mother cell. So it's an asymmetric division. Mother cells and daughter cells behave differently, and you have to keep track of them differently. So the average cycle time of mother cells is 87 minutes. The average size of daughter cells is 112 minutes because they start smaller, they got to grow more. It takes them longer. But the size at division is about 68 femtoliters for both mothers and daughters. Those are the averages, but look at the coefficients of variation. The coefficients of variation are on the order of 15 to 20%. That's life. This is the kind of noise that we have to deal with. Here's a typical histogram for time spent in the G1 phase of the cell cycle. The mean is 16 minutes, but the coefficient of variation is huge, 48%. That's because of the asymmetric division. For the duration of the budded period, the mean is 74 and the CV is 19%. Now here's a neat experiment, again done by Cross's lab at Rockefeller University. They labeled the cells with fluorescent proteins, 2 fluorescent proteins. First of all, a red fluorescent protein was driven by the actin promoter, which is a constitutively active promoter. So these cells are all the time making DS red, and they get uniformly red. And the red fluorescence is a measure of how big the cell is. The bigger the cell is, the more DS red it has, the more red fluorescence it has. So you can measure size by measuring the red fluorescence. The protein myosin 1 is tagged with green fluorescent protein. And the interesting thing is that myosin 1 appears at the neck between the mother cell and the daughter cell in the bud. When the bud first appears, you get a green dot. And the green dot sits there until the bud separates from the mother cell, and then the green dot disappears. Cool. So the green dot tells you when the bud was made and then when the cell divided. And now you can just go in and measure these cells, count the amount of red fluorescence and see when the green dot is there, and you get a very precise measure of the time that the cells are in G1 phase of the cell cycle. That's the time when they don't have a green dot. They're in the G1 phase of the cell cycle. And when they decide to leave G1 and go into DNA synthesis, they also make a bud and they conveniently make a little green dot. So now they've made measurements on lots and lots of cells, and they asked, how does the time spent in G1 phase of the cell cycle correlate with the size of the cell at birth? And I won't tell you why they did this, just take my word for it. It's an interesting plot. There's a lot of variability. Every one of these dots is a single cell that was measured, and measured these two properties. There's a lot of scatter in the data, but if you use your imagination, there are two straight lines with a steep negative slope and a less steep negative slope. Some kind of a statistical fit. And again, this is not the fevered imagination of a theoretician. This is the fit that the experimentalists justify from their data. I'm going to come back to this picture, which is why I showed it to you. What does the model look like? The model was built by these two postdocs in my lab, Devashish Parekh and Sandeep Kaur, in conjunction with a bunch of other faculty members at Virginia Tech and at VBI. So here's the model. A stochastic model has to be a lot more complicated than the simple deterministic models that I was telling you about, because you have to make accurate calculations of molecular fluctuations. So you need molecular details. This model has a lot more molecular details, but you don't have to worry about the details. Just see that it's built along the same principles. This bit is the bistable switch, the antagonism between CLID2 and CDH1. This bit is the negative feedback loop through CLIN2. This little bit is the cell size control that we need to simulate the experiments. And this bit here is the negative feedback loop that activates CDC14. Okay, so it's exactly the same model, but just phrased in terms where we can do stochastic simulations. But we can also simulate the system deterministically. So first I'll simulate it deterministically. There are 58 species, 176 reactions, 68 kinetic constants. Every reaction is described by mass action kinetics. It's an asymmetric division, so the mother cell gets 60% of the size and the daughter cell gets 40%, and that's what a deterministic simulation looks like. No noise, but you're going through the cell cycle, everybody's going up and down just like they should. That's the deterministic simulation that I described earlier. But now we're going to simulate this stochastically with protein populations on the order of thousands of molecules per gene per cell, mRNA populations on the order of 10 molecules, and mRNA half-lives quite short, 2 minutes instead of 20 minutes. And this is what a stochastic simulation looks like under those conditions. There's a manageable amount of fluctuations in the protein levels, and I'll show you that it fits the data quite nicely. But I should back up and say there's a problem here, because we had to assume around 10 molecules of mRNA per cell instead of 1 molecule. It's impossible to make this work with 1 mRNA molecule and a half-life of 20 minutes. It just will not work. We need 10 molecules on average of mRNA per cell, and much shorter half-lives for some particular mRNAs. So these are really predictions of the model, that the numbers in the web are wrong, and they need to be remeasured, and as a matter of fact, we're trying to remeasure these mRNA transcripts in yeast cells. That's the work in Jean Bacot's lab with David Ball. So there's the simulations. Now how does it fit the data? So these are just simulations, and I'm comparing experimental data from the Nature paper with the model simulations, and just look at the coefficients of variation in bold. They all agree quite well with the experimental data. This is the worst one. It doesn't bother me too much, but all the others are right on. We get the right amount of noise in progression through the cell cycle. Here are histograms of time spent in G1 for mother cells and daughter cells. The model on this side, the experiments on that side. That looks pretty good to me. Here's this correlation between time spent in G1 and the logarithm of the size of cell at birth. And this is in the simulation. You can simulate thousands of cells. And here's a kind of fit to two straight lines. And if we compare the numerical simulations with the data from the Nature paper, it's right on. It's the same kind of correlation. The correlation is there, but there's a lot of noise. Okay? Where the underlying determinism of the system is giving you the correlation, but the noisy molecular environment is giving you all the scatter in this data. I hope I've shown you that cell cycle control in eukaryotes can be framed as a dynamical system that gives a coherent and accurate account of the basic physiological properties of proliferating cells. I've shown you that for yeast cells. We also know for lots of other cells. Works very well in understanding their cell cycle properties. The control system seems to be operating at the very limits permitted by molecular fluctuations in yeast-sized cells. And a realistic stochastic model is perfectly consistent with detailed quantitative measurements of cell cycle variability. And that's the end. There are a number of publications that you might be interested in looking at if you want to follow up on what I've described. Thank you very much. I guess I'm taking questions. Yes?
Is there any way to test robustness of this model? Have you stressed it at all to see if it comes back to normal?
Okay, yeah, that's a very good question. And we have done that, but always in the context of what experimentalists do. Okay? So experimentalists are stressing yeast cells all the time, and there are many ways to stress them. The best way to stress them is by introducing mutations into the control system, overexpressing or deleting different genes. And there are hundreds of mutants that have been described in the literature where the cell cycle control system has been perturbed genetically. And the mathematical model that I've described is almost perfectly consistent with all of these experiments. I would say it's 95% consistent. There are some mutants we can't account for. But you can also perturb them by nutritional shifts. There's different ways to tickle cells. And, you know, we're all the time testing our model against those perturbations. So what I would say is our model is as robust as a yeast cell and no more robust than a yeast cell actually is. Yes, Felicia?
So, in the deterministic model, the experiment, the cells are synchronized at the beginning of these cell cycles, is that correct?
Yeah, this is called, what is it called? Induction synchrony.
Yeah.
Because you use mutations to trap cells at a certain stage in the cell cycle. So they're synchronized as far as the cycle is concerned. But they're all different sizes.